So we'll start the class. The topic is the part 2 of immunology. And today we're going to talk about immunoglobulin. But before that, I want to have a recap of the previous class. Question number 1. How many of you say C? Okay, that's right. Immunology means study of the immune system. Next we have the first line of defense against microorganisms. Okay, I know that these two groups of people are very active. I want to hear from the far left, to my left. Please tell me the answer for question number 2. B. The first line of defense is the innate immunity. Then, please this part, the next one, which cell type is known as the antigen presenting cells. The others please don't answer, okay? We'll come group wise. Neutrophil, natural killer cells, dendritic cells and eosinophils. Antigen presenting cells. They don't kill them themselves, but they present the cells to the T lymphocytes. A? All of you agree A? Okay. The answer is C. There are 2 antigen presenting cells, not only 2 but mostly 2. The main 2 are macrophages and dendritic cells. Okay, that's fine. The next one. MHC molecules functions. Any choice? Present peptide antigens to T cells. Anyone else wants to differ from this? All B? Okay, you're correct. It presents the peptide antigen to the T cell. It is inside of the cell, takes the peptide from inside of the cell or outside of the cell and presents it on the cell surface of the APCs and of the B cells. Okay, next, this side. Which of the following is a component of the innate immune system? Is the question visible? B? All agree with B? So the answer is B, because there is no memory for innate immunity. There is no antibody generation for innate immunity. It's a part of humoral immunity, which is a part of the adaptive immunity. Question number 6. To the far left, to my far left again. The complement system. Question number 6. So those of you are saying B, they are correct. It is not only a part of the innate adaptive, it is also part of the innate system of immunity. It is not identical to antibody. There is no antibody generation, and it's not only effective inside the infected cells. So the only correct option we have over here is the serum protein complex of serum proteins that destroys the microbes. So we'll go to today's topic, which is immunoglobulin. So immunoglobulin, there is a structure of immunoglobulin, and this can come as a short note. One question comes from immunology, and usually it is this question: what is the structure of immunoglobulin? Immunoglobulin or antibody, they are one and the same. So we learn about the structure, the function, different classes of immunoglobulins today, and also recognize some of the disorders that can happen because of immunoglobulin deficiency or because of its increment in our body. What is immunoglobulin? Immuno and globulin. Immuno meaning it is related to the immune system. Globulin means it's a protein. It is a glycoprotein. What are glycoproteins? It is a complex of protein and a carbohydrate, right? So that is a glycoprotein. So immunoglobulins are not only just proteins, they are also made up of carbohydrates. So it's a combination of carbohydrate and a protein called glycoprotein that is produced by the plasma cells. Plasma cells are produced by B-lymphocytes, and these are produced in response to antigen. If you remember, humoral immunity, whenever there is an antigen in the body, an antibody is generated against that specific antigen. So this is the antibodies. So if they ask about antibodies, you can write about immunoglobulin. So immunoglobulin antibody, they are one and the same. It is a gamma globulin. Globulin, if you remember, or glycoprotein, if you remember, these are the proteins that are complex proteins. If you remember from the protein chapter, there were two types of proteins. One was simple protein, one was complex protein. Simple protein was just the pure protein part, and complex protein was protein and a non-protein part. So this is a glycoprotein, which is a complex protein. It is also a type of gamma globulin. There are different types of globulin. Globulin, if you remember from the chapter of protein itself, globulin was a globular protein, right? The shape was like a globe, that was opposite to the fibrous protein. So anyway, so proteins are gamma globulin. You don't have to know what is a gamma globulin and everything, but just remember the name. And they are a key component of the humoral immunity. When you say humoral, always remember antibody-antigen production, okay? Antigen-antibody interaction. And they are present in both membrane-bound form and in secreted form. This is the basic structure of immunoglobulin. What is this shape? What do you think it looks like? The letter Y. So remember, this is very easy. There are 4 polypeptide chains. This is 1, 2, 3 and 4. Okay. And what are these? Disulfide bonds. So what amino acid are you expecting to be here? Disulfide bond is seen in which amino acid? There were 2 amino acids where sulfur was there. Cysteine and methionine. Over here we have cysteine, basically. You can see the disulfide linkages are between the 2 different chains and between the same chain. This is one chain, okay? This is not 2 different chains. Intra-chain and inter-chain disulfide linkages. These are called the heavy chain. The longer chain is called the heavy chain. The outside wala chain is called the light chain. So 4 polypeptide chains, 2 types of them. One is the heavy chain. Here also this is the same thing. This is the inside longer part is the heavy chain. The outside part is the light chain. And they have disulfide bonds linking them. And it is Y-shaped. Each of the heavy chain and the light chain has 2 domains or 2 regions in them. One is called a constant and one is called the variable portion. So these portions, this is the constant portion, this is the variable portion of the light chain. Over here you can see this is the variable region. So the tip part are the variable regions of the light and the heavy chain, and the rest of them is called the constant chain. The amino terminal is the variable region. So V is variable region. This is the name is the variable region. So the variable region is denoted by the letter V. Constant is by the letter C. Easy to remember. So this is the amino terminal. This is the carboxy terminal. So the amino terminal of both the heavy chain and the light chain are known as the variable region. VH means variable region of the heavy chain, VL means variable region of the light chain. Same with CL and CH. CL means light chain ka constant region, and the heavy chain is denoted by the letter CH. Letters CH, which is the heavy chain constant part. So you will see, if you divide this whole heavy chain into quarters, so the first quarter is the variable portion, the next 3 are the constant regions. Here half of the, and in the light chain, half of the region is the variable and half is the constant. It can go on, there could be C4 also, CH4 also, that doesn't matter. When you make the diagram of immunoglobulin, make sure that you make at least till 3, CH3. This here you can see denoted by black portion, this is known as the hinge region. which joins the CH1 to CH2. So over here you can see in the first, here it is written antigen, this round circle denotes antigen, and these are the antigen binding sites. So you remember, whenever there is the presence of antigen in the body, our B cells produce, plasma cells will produce antibodies. So antibodies will go and bind with the antigen. That is what we have read so far. So how do they bind? Where do they bind? They bind in these variable regions. Here is where the antigen or any kind of protein or whatever that is not of the body comes and binds. So this is a very important portion of the immunoglobulin. It acts as an acceptor. So the antigen will not go bind over here or over here. It binds here with both the heavy and the light chain. So that area is also known as the antigen binding site. You will keep on reading about it later on in over the course of 5 years. So the hinge region that I have talked about, it provides flexibility between the arms and the stem. So this, you can call it the arm, this part, and this is called as the stem or the base. This is the hinge region, so it kind of provides the flexibility between these two regions. Then we have something called proteolytic cleavage. So whenever we use the word lysis or lytic, it means breakage. Okay, so proteolytic means anything that breaks proteins. Cleavage also means protein breakage. Okay, so papain is one enzyme or one protein that can cleave or break immunoglobulins into two fragments. One is called the FAB and one is called the FC. FAB means fragment antigen binding and FC means fragment crystallizable portions. They have their own importance. We're not going into details about what is the importance of each of these arrangements. But remember that papain is one enzyme that can break this immunoglobulin into two parts over this region. Okay, it will act here, it will break this into two, and you can see here, this will be this portion, upper portion will be known as the FAB, and this region will be known as the FC region. Then now that was the basic structure of immunoglobulin. Do not, if when you are asked about immunoglobulins or structure of immunoglobulins, please make sure that you have these diagrams in hand. Always draw them. Let's understand the function of immunoglobulin. You know by now that they are used to kill microorganisms or any antigen that is there in the body. They cause neutralization, which means that they bind to the pathogens or whatever toxin that are in the body, and they do not let it interact with the host cell. Host means us. Okay, so ours, it doesn't, so the defense mechanism, the antibodies in our body will not let these outside antigens interact with our cells. Then it also causes precipitation or agglutination. When the antibody comes and binds with an antigen, whenever the antigen enters into the body, there will be an antibody that will be released for it. It will go bind to it and make complexes with it. Okay, it will join together and will make certain different complexes, and those complexes can be eaten up by the macrophages, can be easily cleared off our circulation. This is another function of immunoglobulins. Opsonization, you already know. Opsonization means they make a coating. So it coats the microbes and make it more palatable for the phagocytes or the macrophages to eat them. We can clear them off our circulation. That happens through the FC receptor. So this is one of the function of FC region as well. Then they always activate the complement system as well. There are different pathways of complement system, alternate pathway, there is classical pathway, and there are other pathways as well, but it activates the classical pathway. So these are some of the functions, as well as antibody dependent cellular toxicity. So when the NK cells or the natural killer cells, they recognize the FC portion or over here also you see, this is through the FC portion, and here also FC portion. So this is one of the function of the FC portion of the immunoglobulin as well. So the natural killer cells recognize the FC portion of the immunoglobulin that are bound to the target cells, and then they will help them in killing that particular cell or the microbes that are inside the cell. Usually NK cells will directly kill the cells. Then placental transfer of IgG. So IgG is a class of immunoglobulin. We'll go into details now. This will transfer, so placenta is a tissue that provides nutrition to the fetus when it's inside of the mother's uterus. So it forms a barrier between the mother's circulation and the baby's circulation, the baby's body, fetus body. But there are certain things in the body that can cross this placental barrier, like these immunoglobulins, some of the antibodies, not all the antibodies, some of the antibodies can cross it, which includes IgG, and they will give immunity to the growing fetus as well. So we'll go into the classes of immunoglobulins. There are different classes. 5 of them are the most important: IgG, IgM, IgA, IgD, and IgE. They are the names, okay? So we'll learn about IgG first. So this is the main antibody that is present in our body. So in our circulation, in our body, it comprises about 70 to 80% of all the immunoglobulins that are there. So they are the most abundant immunoglobulins, and they can cross placenta. And because it can cross the placenta, it can give immunity to the fetus or the newborn for that. It activates the complement system. And IgG has its subclasses: IgG 1, 2, 3, 4, and IgG3 is the most potent, and it also causes neutralization of toxins. We have already learned about that. This is the structure of IgM. So you see, this is a little different. It's a pentan is the biggest molecule among the immunoglobulins. IgG was the most abundant. IgM is the biggest one because look at the structure, right? They are the first antibody that is produced against any antigen or any infection. So if we have to know, when we want to find out if the person has an infection, if it is a recent infection or if it's a chronic infection, acute or chronic. Acute means recent, and chronic means it's been going on over a number of years or number of months. So what we do is that we check their antibody levels, we check the immunoglobulin type. So if we want to know if it is a recent one, we will find IgM because they are the first antibody to be produced and they are cleared off the circulation very quickly. If we find IgG, it means that this specific infection or inflammation has been going on for a long time. That's how we can differentiate between them. Okay, so if we find IgM, we know that this is a recent infection. You will learn about it more in medicine and in probably microbiology as well. Especially seen in hepatitis. So if we want to differentiate between hepatitis, see if it is acute or if it's chronic, we can do this. So IgM will be produced. It is the first antibody, and it has no memory cells, and it is very potent. It is present on the B cell surface. B cell means B lymphocytes and acts as an antigen receptor, which means receptor means it's for receiving the antigen, and act as opsonin as well. IgA is present in all of our body secretion. It is present in our tears, it is present in the saliva, so, and all the other body secretions as well. Whenever you think about secretion, think about IgA. It is the second most abundant antibody after IgG, and it is effective against bacteria, against viruses. Anybody can tell me, what does Vibrio cholerae do? What is it causes which disease? Which disease? Cholera. Neisseria gonorrhea? Gonorrhea. It's the name itself, polio and influenza viruses. So it's effective against viruses as well as bacteria. This is present in the breast milk as well. That's why mothers, when they breastfeed the baby, they will be able to transfer the antibodies from the mother to the baby and help in the in making the immunity of the baby better. This is IgD. You don't have to remember each one of those structures, okay? You have to remember the basic structure of an immunoglobulin. This is IgD. This is present in very low concentration. It is also found on the cell surface of B lymphocytes along with the IgM, and they are involved in B cell activation. There is no other function that we have seen so far for IgD. Then we have IgE. This is also present in very low amount, but it is very important. It is very, very potent in action. It is responsible for immediate hypersensitivity. You will see allergic reaction. If somebody is having allergy to the, let's say, egg, you will see that immediately after eating egg, they will have itchiness, or if somebody is allergic to seafood, they'll have itchiness, they'll have swelling, sometimes they'll not be able to breathe, and all of those things happen because of this antibody, IgE. It is seen in asthma. Those who have asthma, you see people have this reaction very quickly. This is because of IgE. You can see this in anaphylaxis. Anaphylaxis is severe form of an allergic reaction. Some of the people, you may have heard that if they are allergic to something, or let's say a drug, it can be any food allergy as well. So if some of children have peanut allergy, so whenever they have peanut, what can happen? They'll have sudden, it will not just be itchiness and little swelling. So they'll have swelling, or edema is the word we use for swelling. So they'll have edema all over their body. They'll have in their throat, so they'll not be able to breathe. So they'll have difficulty in breathing. They can go unconscious. They can have fever. They can have diarrhea. They can have vomiting. All of those things can happen. This is severe form of the allergic reaction, which is called anaphylaxis, and that is life-threatening. Person can die within minutes as well if we do not treat them properly. So this IgE, you can imagine IgE is doing that. That means it's very, very potent. It is also seen in Helminthic infection. Helminth means worm infection. So if you take a person, he's having whatever sickness. So whenever we check any person who comes to the hospital, we're always checking their blood reports, right? So we're gonna check their blood. We're gonna check their peripheral blood smears. We're going to make slides, and we will see the types of cells that are there. We'll see if there are parasites. We'll see these as well. So if IgE is there, that means it is showing an allergic causation. So we can think about, so this will help us diagnose the patient's condition as well. So we can pinpoint which condition the person may have. They are present in basophils and mast cells. Basophils and mast cells are responsible for release of certain granules from them that causes this reaction. These are certain disorders that are associated with immunoglobulins. If you see the first one, we have IgG. So disorders that are related to IgG, we can have Hyper-IgG, where there is too much production of the IgG type of immunoglobulins that are seen in chronic infection, autoimmune disorders. I think class has been taken on autoimmunity in physiology, right? If not, you will get it. Autoimmunity is when our immune cells, which are responsible for taking care of our body, will turn against us. So for some reason, our own immune system will start attacking our normal cells. You cannot differentiate between the foreign cells and the normal cells anymore. That leads to autoimmunity, like rheumatoid arthritis. Then we can also have conditions where there is deficiency of this immunoglobulin, and because this immunoglobulin is deficient or not sufficient in numbers in the body, it will lead to recurrent infection. We'll always have infection, sardi, khasi, everything, GI problems, respiratory tract problem. They're involved in this. And when we come to IgA, there is something called Selective IgA Deficiency. All the other immunoglobulins will be okay, but the IgA will be deficient, and so the person will be very susceptible. Susceptible means they'll be very prone to developing the same thing, like recurrent infection, GI and respiratory infection. GI means gastrointestinal infection. There is something called IgA nephropathy as well, which is a kidney disease, a common kidney disease where there is deposition of IgA in the glomeruli cells of the kidney. So that will lead to kidney damage. Then there is something called celiac disease. Somebody was asking me what is celiac disease. So here we have gluten that is present in bread and other food stuff. Person is allergic to that, or when person is allergic means what? There is an immunoglobulin antibody produced against gluten. So this is called celiac disease. So person will have GI upset, they may have itchiness and all of those things. This is an abnormal reaction. Then we have IgM, which is called as a Hyper-IgM Syndrome, which is a genetic disorder where the hyper means more, right? Increase. So hypo is less. So whenever there is hyper-IgM, means there will be a lot of IgM in the body and very low the rest of the other immunoglobulins. IgD, myeloma. Myeloma is a plasma cell malignancy or the cancer of the plasma cells. This is called, so this can happen in IgD, and also hyper-IgD syndrome, where you might have a periodic fever here and there, every time there is a fever with no infection, and they can have auto-inflammatory, meaning that there is inflammation because of our own immune system. IgE seen in allergic disorders, you have already seen. So I am not covering here multiple myeloma, which is the where there is cancer of the plasma cells, where there is a lot of immunoglobulin production. This will be taken probably your practical classes when you talk about proteins, or maybe even in the urine. There are Benz-Jones proteins and all of that. That's part of, or maybe we will take it up as an ECE. So for today's class, I'm not taking that. These are all the disease that you can mention if it comes in your exam. So this was a short class.